Yeah.
So what's the difference between lenti and retroviruses again? Like,
Oh, I'm sorry. Lentiviruses would include is a type of retrovirus.
right.
So there's spumaviruses, there are lentiviruses. Those are all viruses within the retrovirus genus, retrovirus group, retrovirus family. And lenti means slow, and so the slowly developing virus. And so HIV would be in there, and also HTLV is in there as well. Good question. Let's go then to vaccines. We're going to start with attenuated vaccines. And with attenuated vaccines, we are going to have a vaccine which has been normally passaged many times. I had a neat picture of them passaging it. Let me go back up. Let me see if I can type in passaging. Well, I had that picture and I'm not sure where I got it. Okay. We'll go back. But basically with attenuated viruses, attenuated vaccines, what you want to do is you want to be able to, the way these are made, that have been made, for example, the Sabin vaccine was made by taking a live polio virus and a fully infectious polio virus and passaging it through a series of other hosts, through monkey cells, for example, and passaging it until it became evolutionarily adapted for the monkey cells it was passaging in. And it evolved away from human pathogenicity. And this is a common thing with viruses. So for example, when I was doing my doctoral work, one of my mentors was passaging, had isolated a virus that grew very well in lettuce. And he grew it on, it was a lettuce strain, and he grew it on tobacco, because that's commonly what we grow it on. And he grew it on tobacco for half a year, passaging it regularly. And then he tried to put it back onto lettuce. It would no longer go onto lettuce. It just had lost that ability. So that is an idea of passaging. If you passage it in a different host, it's going to lose it. And so it's very common with plant virologists is they might passage it in tobacco to build up stock, but they make sure they come back and passage it to the original host, otherwise they're just going to lose it. The virus will evolve. So the same thing happens with the polio vaccine. You passage it into another host, it will evolve away from the human. So these are called attenuated vaccines. There are other ways that you can make attenuated vaccines, much trickier, and that would, one example would be you start modifying with the polymerase, so you might start modifying with some other protein of the virus. You might mutagenize it, and that way it just doesn't replicate as strongly as the wild type. Or it just doesn't interact with the immune system quite so strongly to cause some sort of pathogenicity. Because actually you can have an overreaction with the immune system, or with the wrong arm of the immune system, like with Th1 rather than Th2, and that would be the source of the pathogenicity. So if you use your head, you can rationally change viruses and attenuate them that way as well. But the random way of doing it is passaging them through another organism. once you have it passage, then you can do some neat stuff. And so an example here would be, here you can take an attenuated flu strain, for example, and then you have a brand new flu strain which is really causing a lot of troubles. You can go ahead and selectively come in and take just a couple of components. So flu actually has 8 RNA segments to it. So you can take 2 of those segments for HA and NA, for example, and you can pop it into the attenuated donor strain, and you might very well have an attenuated strain which has the new HA and NA, the hemagglutinin and the neuraminidase. These are the surface proteins for the virus. So now we started off with our attenuated donor strain, and we have replaced the surface proteins with the surface proteins of the virulent strain, and now we are able to have an attenuated strain which can be recognized by our immune system as the virulent strain, but it really is attenuated. So that's another way that you can do it. Okay, those are attenuated viruses. These are going to be live virus vaccines. So you inject it into people, and they actually, the virus multiplies in them. And what are the advantages with a live virus vaccine? Why would it be so powerful as a vaccine? And here are some live attenuated viruses. So polio can be as a live virus vaccine, but we also use it as a killed virus as well. Hepatitis A, measles, mumps, and rubella, varicella zoster, which is a chickenpox virus, and tuberculosis and yellow fever. These are all live virus vaccines which have been attenuated. So you can think about it, I just asked that question, but I'll just answer it real quickly, is the full panoply of the immune system is activated because we have a virus which is replicating. So the innate immune system immediately launches, and it tells the adaptive immune system, which is the T cells and the antibodies, the B cells, hey, we have a problem here and we have to get going. Okay, let's look at a different version here, and we're going to talk now about killed vaccines. And in this case, you're going to come in and take a virus and that doesn't do it. Okay. You're going to take a virus and you are going to here we go. Oh, come on. You were supposed to come up. Please come up. There we go. Okay, we got it. So we have some viruses, vaccines, which are live attenuated. Others are inactivated or killed vaccines. And in this case, you take the organism, whether it be a bacterium or a virus, and you treat it so that it's dead. So you might treat it with formalin, for example. And then you just inject it into the person. It's a dead virus. It shouldn't hurt the person, but it has all those antigens there. And I have a question for you. Would a killed virus, can it activate the innate immune system, even though it's dead? Because one thing innate immunity can do is it can see, hey, we have this virus which is replicating, and you can do it. But if we put in, like, say, a killed pertussis or cholera bacteria.
If it's bacterial, would it still have surface antigens?
It would. So we would have the lipopolysaccharides, and we would have the peptidoglycans and other things. And what recognizes those?
Macrophages.
Yeah, macrophages, toll receptors, right? So the innate immunity has the capability of looking at patterns, certain common patterns in pathogens, and identifying them and saying, this is a real problem. And so they can do that in the innate immunity. And then it clicks on the adaptive immunity from there. So yes, killed vaccines are able to do that. So let's move on then. The problem with killed vaccines is if you kill them too much, then you destroy the proteins that elicit the reaction. If you don't kill them enough, then they are virulent, and you're injecting your patient with the thing you want to protect them with. So that's the two sides to it. So which is safer? Killed vaccines or attenuated vaccines? Well, there are pluses and minuses for both of them. Subunit vaccines, here you're going to instead of adding, you could add the entire virus. You could add the entire virus, and your antibodies would bind to these particular antigens. And you could do that with a killed vaccine or an attenuated vaccine. Instead, what you could do is you can inject the patient with just the protein, just the surface protein of that virus, let's say, or the bacterium. And then your antibodies will detect just the surface protein. Now, will the innate immunity recognize this? Well, maybe not, because if it's just the surface protein, if only one protein, you're not putting in liposaccharide, you're not putting in alpha-1,4 galactans, you're not putting in anything that the innate immune system has recognized. It recognizes patterns, not particular proteins. So that's the big problem with subunit vaccines, is you have to include something that would stimulate the innate immune system. What do we call that something we add to stimulate the innate immune system?
Did you get it?
No.
No.
Close. Begins with an A.
Adjuvant.
An adjuvant, exactly. The only problem is the adjuvants approved for human use are basically aluminum phosphate salts and not too much else. I think Q21, which is a saponin, it's a type of detergent from a plant. So very little to stimulate this. And so other things like Freund's Complete Adjuvant and others are not allowable because they can give bad reactions. So that's the problem with subunit vaccines. But still, there are subunit vaccines. For example, hepatitis B is a subunit vaccine. All right, let's look on then a little further, and we're going to look at DNA vaccines. So here, you just say, well, you know, we have the clone for this subunit vaccine. Why go to all the trouble of producing protein when we could just, why don't we just inject the DNA? That sounds good. Doesn't that sound good? Let's just inject the DNA. And so let's see how this works here. So what you do is you just inject the DNA. And this sounds like the stupidest idea in the world. Load up a syringe, inject the patient with DNA. But what happens is you inject it usually intramuscularly, and the DNA finds its way into these cells, and it produces protein in the cells. And then the protein produced in the cells is loaded up into the major histocompatibility complex, and then the CD8 cells come in and recognize that there's a problem. And of course, you could also do it with, if they get into dendritic cells, and you can actually activate then these cells to raise the alarm call that we have some problem which is occurring there. So getting it into muscle cells or getting it into dendritic cells of some sort, the DNA actually works. Now, interestingly, is when you inject a subunit vaccine, you're injecting milligrams of protein. When you inject the DNA vaccine, the total protein production is so minuscule, it's in the nanograms. And yet the reaction can be just as good as milligrams of protein. The reason being is that when you make a subunit vaccine, you have to make it in something else, like plants or yeast or bacteria, some other vector. And it never is folded exactly quite right. It's never glycosylated exactly quite right. With the DNA vaccines, it goes in there, and it will then be produced in the human cell of that patient, in the appropriate human cell of that patient, perfectly folded, perfectly glycosylated, expressed in all the right components, loaded up into the major histocompatibility from the inside. Everything's perfect. And for that reason, you get a very strong response. The other great thing about DNA vaccines You can store DNA at room temperature, as long as there's no DNA in the solution. You don't have to freeze it. It's fine. I know a lot of people, I know one person at least used to store his plasmids right on his desktop. As long as they're clean, they store almost forever. Now, the next thing, the final thing we're going to look at in this lecture today is virally vectored vaccines. And in this case, what you do is you have a recombinant viral vector. And this gets into, say, the dendritic cells, right? And what you do is, it is just a viral vector that replicates in humans. You can pick anything you want, adenoviruses, AAV, anything. As long as it replicates in humans and doesn't cause too much damage. So alpha viruses can do this. There are special strains that don't cause apoptosis. So that's another one you could use. The patient gets this innocuous infection with the virus, and the virus hopefully gets into the dendritic cells, and then it expresses the protein. You put in the virus is a carrier for your vaccine protein. It expresses the vaccine protein in very high quantities. Is the innate immune system turned on? Yes, it is, because, of course, you have the virus is multiplying, so the innate immunity is turned on. And you express this. You could express it in dendritic cells or in other cells. And suddenly, you have the protein produced at very high levels. The innate immunity is turned on, and everything works well. So you could even use this for cancer vaccine. So here we have TAA, the tumor-associated antigen. You could be expressing that at a high level and have a cancer vaccine, for example. So that's all we have for today. And therapeutics and vaccines. So.